Okay, welcome everyone to today's Immune Zoom seminar. It's my great pleasure to introduce our speaker, Dr. Masanori Aikawa, who is currently the professor of medicine at Harvard Medical School and the Yoshihiro Miwa Distinguished Chair and the founding director of the Center for Interdisciplinary Cardiovascular Science at Brigham and Women's Hospital. Masanori received his MD and PhD at Juntendo University in Tokyo. He came to the United States in 1995, starting as a research fellow in Brigham and Women, and was promoted to assistant, associate, and full professor. His studies focus on macrophage activation in atherosclerosis, obesity, insulin resistance, and fatty liver. His laboratory used various in vitro and in vivo approaches, including macrophage selective knockout and transgenic mice, and molecular imaging of atherosclerotic plaque macrophages. Masanori has made numerous important discoveries in vascular biology. For example, he provided strong evidence to support the plaque stabilization hypothesis. Together with Pete Libby, Masanori pioneered the concept that lipid lowering is an anti-inflammatory therapy and providing molecular basis for the observed beneficial outcomes of statin. He also firstly demonstrated in vivo the role of MMP collagenases, the major macrophage products, in plaque matrix remodeling. And more recently, Masanori's lab has explored the new mechanism that trigger macrophage activation, including the delta-like 4-notch axis, the AKT beta-catenin pathway, and the pro-inflammatory action of apolipoprotein C3. And because of these exceptional contributions to science, Masanori has received numerous awards, and including the Special Recognition Award in Vascular Biology and the Jeffrey Holt Award from American Heart Association. He is also an overseas fellow of the Royal Society of Medicine in UK and an honorary member of the Korean Stroke Society. Today, Masanori is going to talk about a system approach to target discovery for inflammatory vascular disease. Without further ado, Masanori, the floor is yours.
Thank you. Thank you very much, Dr. Su, for your kind invitation and also introduction. I'm so excited to have this great opportunity to share with you our interest in macrophage biology, and then we use systems approach, as the doctor said, Su said, for target discovery research. Okay. Okay, I cannot. Oh. I'm sorry, I'm struggling to advance my slide.
Okay, so can you use your keyboard? Does that work?
Yeah, I'm using that.
Okay, so maybe change back to the regular mouse.
Okay.
Because sometimes the Zoom has some. Okay. So
okay. All right. Sorry about that.
No, great.
Okay.
Great.
Yeah. So we are interested in macrophage activation because no matter where they come from, macrophages play an important role in various vascular diseases such as atherosclerosis. And also, I'm going to talk about the vein graft failure a little bit later. Okay, this is how coronary artery looks after myocardial infarction. This coronary artery, was completely occluded with thrombus that killed this patient. Why this occurs, and then what what roles macrophage play. So that's the story of my talk today. So question is again, why acute myocardial infarction occurs, right? But this is the very interesting series of discovery from angiography study. And then in late 1980s, early 1990s, they reported that most acute thrombotic event occur in coronary arteries that did not have, critical stenosis previously, which means that plaque size does not matter. If plaque size doesn't matter, what matters? For example, this patient, coronary artery, this artery, right? It's quite intact, but this can get occluded like this. Again, what matters? What causes this? If size doesn't matter, probably quality matters. So that is, the discovery of, some investigators in 1960s and 70s. For example, simply speaking, that if the coronary lesions contain so many macrophages, they crack. That was called plaque fissuring, but later it was commonly called plaque, rupture. Then, the characteristics of such, kind of rupture prone region is they are rich in lipids and rich in macrophage form cells and poor in collagen. Then I'm going to talk about the balance between inflammation or macrophage accumulation, then collagen. Right. Okay. In mid-1990s, Peter Libby and I proposed that macrophage-derived MMP collagenases degrade fibrillar collagen, which is a molecular determinant of plaque stability. Like fibrillar collagens are something like this, almost like a cable of a bridge, right? But if you have macrophage-rich plaque, and that they are vulnerable, and because of expression of MMP collagenases by macrophages. That's the theory. I can skip this. Okay. And then early 2000, we used collagenase resistant mutant knock-in mice and collagenase 3 or MMP13 knockout mice to show that MMP collagenases indeed regulates collagen accumulation and structure in atherosclerotic plaques, providing the first direct evidence for the role of MMP collagenases in plaque stability. So what causes macrophage accumulation in arteries? So now we need to go back to very old study, in early 20th century, Russian pathologist Nikolai Anichkov, used fat rich diet. Actually, that he fed rabbits eggs and then identified the lots of macrophages in atherosclerotic plaques in rabbits. So lipids are bad. Then, when I was a fellow in the Brigham Women's Hospital with Peter Libby, we used the rabbit models of atherosclerosis to show that cholesterol diet by either diet or statins can decrease the macrophage accumulation or activation, which kind of provided the molecular basis for the so-called plaque stabilization hypothesis that lipid lowering therapy. Basic science, evidence, was fully supported by multiple statin mega trials, such as this Jupiter, led by our colleague, Brigham Women's, Paul Ritker. So we learned a lot from these studies, and the statins can save patients. However, we also learned that statins reduce cardiovascular events in many, but not all people. Therefore, my laboratory has been focusing on identifying new mechanisms for macrophage activation as therapeutic targets beyond modifiable risks such as hyperlipidemia. However, I also recognize the major challenges in drug discovery research, First of all, target discovery takes long time. And also, probably a more problematic challenge is that success rates of new drug targets that are often identified in academic labs in future clinical development are extremely low. So what causes these challenges? One problem may be that biological systems are much more complex than we traditionally thought, right? A causes B, then B causes C. So therefore, we thought B inhibitor can solve, all the problems, and then, kind of some disorder C or activation of molecule C, wouldn't occur. But biology is not that simple. That's what we learned, over 10 years ago. Such signaling, mechanisms may be buried in the very middle of complex biological network. We therefore decided shifted over 10 years ago from hypothesis-driven research to systems approach, which involve unbiased omics and bioinformatics that can speed up the process. And also network analysis to predict clinical impact, which ultimately increases success rates in clinical trials of new drugs. Then, in systems approach, we believe that network medicine and multiple omics integration are very important, particularly for complex cardiovascular, diseases. In my group, each study typically begins with omics screening. Could be proteomics, could be RNA-seq or single RNA-seq. The samples could be, cells or animal tissues or clinical samples. And then recently we are using EHR data as well. Then, such data, are usually big, and then they undergo bioinformatics program. Then more importantly, this process is followed by computational prediction. This is the kind of solution, to the challenge two I mentioned. The success rates of drugs in clinical trials are extremely low. And then more importantly, after the unbiased screening and then target identification, it is still very important, for you to perform in vitro, in vivo validation. However, our model is before we spend too much time and effort to really make sure that your target works, we will start thinking about drug design. So that's the new model. In order to establish the systems approach to target discovery, we have quickly implemented the multiple innovative technologies such as proteomics, systems biology, and also some bioinformatics methods for large biological data. Actually, we have developed our own methods. And also, we started using computational drug screening method. And then more recently, we started using AI. So why innovative technologies are important? We believe they increase the speed and thus reduce the cost ultimately. And also, more importantly, innovative technologies support new paradigms. And systems biology, the use of systems biology was encouraged by Dr. Gary Gibbons, director of NHLBI in 2016. He's actually a former colleague of Brigham Cardiology. And so this is a very important trend, I believe. And then to really identify the promising targets that can survive very challenging clinical trials. So I run cardiovascular research at 2 labs. One is in the Center for Excellence in Vascular Biology in this building. And then in the next building, I direct the Center for Interdisciplinary Cardiovascular Sciences. So this is the former fellows and then our recent members of the our laboratories and run with my wife Elena Aikawa. And so this operation is supported by National Heart, Lung, and Blood Institute or American Heart Association. Then we launched the Center for Interdisciplinary Cardiovascular Sciences, or CICS, in collaboration with Japanese pharma, Kowa, in 2009, as a new model of academia industry collaboration in drug discovery. I'm a founding director, and then Elena Aikawa is a co-director. So now I'm going to share with you, several examples of our study using systems approach. In this study, our former fellow Hiroshi Iwata used unbiased global omics and systems biology to identify new molecular switches of macrophage activation. We used traditional M1, M2 polarization model. And Hiroshi used interferon gamma stimulated macrophages and IL-4 stimulated macrophages as a models of pro-inflammatory as anti-inflammatory macrophages to identify the new regulators of macrophages. Then we identify the PARP9 and PARP14 from this exercise. Then we knew nothing about these molecules, and then learned a little bit about them. PARPs are a family of 17 ADP ribosylation enzymes. PARP catalytic domain transfers ADP ribose moieties from NAD to protein acceptors. Then this process is called ADP ribosylation. Then ADP ribosylation is one of the fundamental post-translational modifications. Then this may contribute to various aspects of normal homeostasis and disease mechanisms. Particularly at that time, there was accumulated knowledge in the role of ADP ribosylation in cancer biology, but roles of PARP9 and PARP14 in vascular inflammation were completely unknown. So therefore, we were very, very interested in this. However, we recognize the potential challenge that PARP9 and PARP14 would interest in the important in macrophage biology, but probably drugs against those compounds may not work in humans in clinical trials. So therefore, we performed computational prediction for their clinical impact by network analysis. First, we made a PARP9 and 14 network with their first neighbors, and then compare this network with human disease gene modules, and then found that PARP9 and 14 network was quite close to human coronary artery disease gene network. I'm sorry, my computer said battery power is low, although it's connected. Okay, I think it's okay now. Sorry about that. So because of this data, we decided to go ahead to work on PARP9 and 14. We performed multiple in vitro mechanistic studies to found that PARP9 promote and PARP14 suppresses macrophage activation via STAT1 and 6 pathways. More importantly, we performed in vivo validation study to found that the PARP14 is indeed anti-inflammatory. When we transferred bone marrow cells from PARP9 null mice to LDL receptor knockout mice, lesion area increased, and then macrophage accumulation also increased. And also we found the increased expression of pro-inflammatory genes in PAR14 knockout tissues. Okay, I'm sorry, I still having the warning from the computer.
Well, yeah, that's fine. Yeah, take your time.
Yeah, sorry about that. I don't know.
No problem. No worry.
I never experienced anything like this. It's flagged. That's strange. Okay. 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 Maybe I'd better move to the. Hold on.
Sure. Yeah, we can move to another computer if it works better in that way.
Okay, it should be fine. All right. I'm sorry. I'm very sorry about that.
Oh, no worry.
Okay. All right. So anyway, in vivo validation, told us that PAR14, is indeed anti-inflammatory molecule as expected from in vitro study. Then, I'm going to talk about the biology of macrophage heterogeneity, in this study. I'm going to show, some data in each study. So as you know, that the M1, M2 dichotomy, is a very popular theory. Then, in vitro studies, told us that there are at least, two subsets of macrophages, and pro-inflammatory M1, or anti-inflammatory, M2 macrophages. But, we also understand nowadays that this is overly simplistic. But, when we identified PARP9 and 14, we thought the regulators of M1, M2 balance, could be the promising therapeutic targets. Yeah, and then, we performed some prototypical singles analysis at that time. First of all, we really want to know, the patterns of heterogeneity of so-called M1 macrophages, and then identified very interesting thing. In this single cell qPCR analysis, we found that even after interferon gamma stimulation, human primary macrophages were heterogeneous. And we found that M1 macrophages are, still heterogeneous, and therefore this theory is really, overly simplistic, and the macrophage heterogeneity is more complex. Then I'm going to show you more data later. So from this study, we identified new targets, and also more importantly, we decided to start working on the new concept. The biology of ADP ribosylation, is important, in inflammatory vascular disease. It was a totally unexpected new direction, that was generated by unbiased screening. And also, more importantly, this new concept kind of forced us to develop new technology, so which is ADP ribosylation proteomics. Okay. And so this is very interesting era. There are so many new technologies. Then, as I mentioned earlier, new technology, helps you develop new paradigms, right? And also, interestingly, new paradigms developing new paradigm further requires additional new technology too. So this kind of synergistic interaction is very, very exciting, nowadays. So we should really enjoy that. Then, as I mentioned, that we realize that we really need to develop unbiased, ATP ribosylation proteomics. Then, in our center, so we have proteomics research group led by Sasha Sin. Then, we published, a few papers already. So this is just a summary of in vitro version of ATP ribosylation proteomics, and this is in vivo version. Interestingly, a liver, ATP ribosylation signatures and spleen signatures are very, very different. Then now, our obvious direction is to establish ATP ribosylation proteomics and atherosclerotic plaques. Now we are working very hard to establish mouse version of ATP ribosylation proteomics, in atherosclerotic plaques. Then, we are hoping to submit a paper in the near very future. Now, I'm going to talk about, chronic kidney disease. CKD, as you know, is a global burden. And then, we are dealing with diabetes pandemic, right? And then, which increases the prevalence of diabetic nephropathy, thus CKD. And then CKD is known to promote inflammation, thus cardiovascular risk. Then what are the mechanisms? We use systems approach to target discovery, to identify the potential regulators of accelerated vascular inflammation in CKD. So this paper was published in Circulation, a few years ago. Then we use uremic toxin, which is a major uremic toxin, indoxyl sulfate, which is a major uremic toxin in CKD patients. As we did in PAP study, this study started with proteomics of indoxyl sulfate elicited macrophages, followed by bioinformatics program, and then pathway analysis. During this process, we identified that NOT signaling was highly enriched in indoxyl sulfate treated macrophages, and then network prediction linked this cluster with human disease modules such as myocardial infarction and atherosclerosis. Then we decided to work on this mechanism related by NOT signaling. Then in vitro mechanistic studies demonstrated that the OATP2B1, which is a family of transporters, contribute to the kind of internalization indoxyl sulfate into the macrophages, which then modulate ubiquitin proteasome pathway, which leads to the dramatic induction of delta-like 4 ligand, which activates NOT signaling, and then M1 macrophage polarization. Then in vivo, validation used macrophage targeted siRNA for delta-like 4 or OATP2B1 that showed that the suppression of either delta-like 4 ligand or OATP transporter substantially suppress the accelerated atherogenesis in mouse atherosclerotic plaques induced by indoxyl sulfate administration. So now, this is another example of macrophage heterogeneity. In this study, we applied indoxyl sulfate to human primary macrophages and then saw dramatic induction of delta-like 4 as early as 30 or 60 minutes. Then we realized at that time, this is interesting, but not all macrophages express delta-like 4. Some delta-like 4 positive cells were negative for a macrophage marker. Oh, I'm sorry. So some macrophages are negative for delta-like 4. And then, what are those delta-like 4 positive cells? are they more pro-inflammatory? So we saw similar phenomena in vivo as well. This is a mouse vein graft tissue. Then same thing here. Not all macrophages express delta-like 4. Then some macrophages were delta-like 4 negative. So what are those delta-like 4 positive cells? Are they more pro-inflammatory? We thus performed a single-cell mass cytometry or cytof of macrophages treated with or without indoxyl sulfate. So this is control macrophages. This spade tree analysis image demonstrate the clusters of macrophages. Then overall delta-like 4 expression levels were low here. However, we also realized that the control macrophages were quite heterogeneous as well. But if you add indoxyl sulfate to these macrophages, delta-like 4 expression levels were induced. So that some cells, some clusters became red. However, we also realized that some cells, some clusters were high responders, some clusters were low responders, which means that the macrophage heterogeneity is regulated at multiple levels. Then going back to the original question about the phenotype of delta-like 4 positive cells, we looked at the expression levels of some chemokines, cytokines, then TNF-alpha expression patterns in this step spade tree analysis from cytof were extremely similar to delta-like 4, suggesting that delta-like 4 expressing macrophages may represent pro-inflammatory subsets. So now this is another example of the use of systems approach. This paper was published in Circulation Research last year. As you know, PCSK9 is a very, very popular target. Then PCSK9 inhibitors are on the market as a lipid-lowering drugs. So PCSK9 promotes the degradation of LDL receptor and regulates cholesterol metabolism. And also, some literature demonstrated that PCSK9 targets other than LDL receptors, such as LRP1 and some others. So we therefore, and also clinical evidence demonstrated that PCSK9 levels were higher in patients with saphenous vein graft disease as compared to patients without saphenous vein graft disease. I'm going to talk about vein graft a little bit later. And also, some basic science study demonstrated that PCSK9 may have pro-inflammatory effects, independent of LDL receptor. So what is the vein graft failure? Because of diabetic pandemic, prevalence of peripheral artery disease, PAD, is increasing. Then vein graft implantation is the very common procedure to PAD. But problem is 30 to 50% of PAD vein grafts narrow or are occluded within 1 year. 30 to 50% initial year event rates are extremely high. The current options are very limited, then economic impact is high. However, no medical therapies are available, probably because mechanisms are still unclear. So we therefore use the mouse model of a vein graft implantation, where we transfer inferior vena cava to carotid artery of LDL receptor knockout mice. Within 1 month, in this model, vein graft developed very complex lesions, almost like advanced human coronary atherosclerotic lesions. And in this study, we used adeno-mediated transfer of gain of function of PCSK9 in LDL receptor knockout mice. So therefore, no matter what you see, that's, LDL receptor independent. Then in this study, we saw PCSK9, accelerated the vein graft lesion development. We also saw the PCSK9 administration increase the macrophage accumulation in LDL receptor knockout mice. So question was, what mediates LDL receptor independent pro-inflammatory effects of PCSK9? Then we performed the RNA sequencing, and then to identify some pathways that were highly enriched in PCSK9 treated macrophages. Then we use some unbiased approach, which is a network-based hyper-edge entranglement prediction algorithm, or HEP algorithm, developed by our colleague. And so this is a very interesting method. We first perform proteomics of cell surface proteins, and then with PCSK9 treatment, some proteins amounts decreased, including LDL receptor. And also, we used the RNA sequencing to show the downstream events, as determined by expression levels of mRNAs. And then basically we sandwich the proteomics of cell surface protein and RNA-seq data of downstream events, and then identify what mediate those changes in upstream, downstream. Then I identified multiple molecules, including Syndecan4, SDC4. We also analyzed the in silico protein-protein docking analysis, and then realized that the binding of SDC4 to PCSK9 is as strong as that of LDL receptor and PCSK9. And then if you silence, SDC4 in PCSK9 treated, macrophages, some of those pro-inflammatory molecules decreased. So this is another study, that used systems approach to target discovery. And in this study, Julius Decano used systems unbiased proteomics, of vein graft tissue from mice. Then we performed the multiple analysis from different angles, but in this analysis, we found during the development of vein graft lesions, PPAR alpha expression decreased at protein level. Then PPAR alpha at that time was already known as an anti-inflammatory molecule, but nothing was known about its role in vein graft lesion development. And also question was, PPAR alpha decrease, so what? Does it have a causal role? So therefore, we performed loss of function and gain of function of PPAR alpha, in mouse model of vein graft lesion development. Then, this is the loss of function study involving macrophage targeted, lipid nanoparticles that contain the PPAR alpha siRNA. So this is the in vivo delivery of siRNA against PPAR alpha, and the result indicated that PPAR alpha silencing, increased the volume of vein graft tissue and also wall thickness. So we also use the PPAR alpha selective, activator pimafibrate, in gain of function study. If you activate PPAR alpha pathway, macrophage accumulation decreases, and also macrophage activation as determined by molecular imaging for MMP decreased as well. And interestingly, Ly6C plus monocytes, which is a subset of a kind of pro-inflammatory monocytes in circulating blood, decreased with pimafibrate, treatment. The PIP-alpha is a regulator of cell metabolism. And also, we had already learned that the M1, M2, polarization, is also associated with a metabolic balance. So therefore, we performed that so many, metabolic and then studies. And if I summarize this data, we found that the PIP-alpha activation by pimafibrate mitigates hyperglycolytic state in LPS-elicited macrophages. So this was consistent with previous finding that the inflammatory states of macrophages are closely associated with intracellular metabolism. Then again, PIP-alpha activation by pimafibrate reduces pro-inflammatory parameters of macrophages, probably through multiple metabolic pathways. So in this study, we also performed a single analysis of macrophages. And so this is control macrophages. This data represent LPS-stimulated or traditionally M1 macrophages. Then as you can see here, as determined by CCL2 expression levels, LPS stimulation, kind of cause the appearance of new island here. Okay, this is consistent, among many parameters. But if you add pimafibrate to LPS-stimulated macrophages, this island disappears or gets closer to the mainland, suggesting that pimafibrate, not only regulates macrophage activation, but also macrophage heterogeneity. So now, I've been talking about the traditional M1, M2, balance, and then PARP9 and 14, and also delta-like 4, PIP-alpha. These guys could be regulators of M1, M2 balance. However, in the last, several years, we have learned a lot about this. M1, M2 theory is overly simplistic. And then there are more subsets of macrophages. So macrophage heterogeneity is a multidimensional rather than, M1, M2 dichotomy. So this is exciting, however, very challenging. As technologies develop, for example, from bulk qPCR, single RNA sequencing, data complexity and size have also changed. Now we are dealing with very complex and huge data. How can we deal with this? The answer could be multiple, but one possibility is the use of AI. Then in this study, we formed the team AI so that we can use machine learning to analyze very complex single RNA sequencing of human primary macrophages. For this study, Julius Decano was a finalist for American Heart Association Irvine Page Junior Faculty Research Award 2021. The currently, paper, a revised manuscript is under review for circulation. In this study, we used LPS and interferon gamma to activate, macrophages or, create so-called M1 macrophages. So this is a PSA plot of control LPS-stimulated, interferon gamma-stimulated macrophages. And as you can see here, MLPS or interferon gamma are very, very different as determined by expression levels of genes, identified by, single RNA-seq. So, although they were both called M1 macrophages, LPS-stimulated, interferon gamma-stimulated macrophages are very different. And if you look at interferon gamma-stimulated macrophage more closely, there are at least, few, subsets. So which is consistent with what we saw, in our earlier study on PARP9 and 14, where we used more prototypical, a single cell qPCR machine. Then if you look at these subsets of macrophages more closely, you can appreciate that cluster 2 cells may be more pro-inflammatory. And then cluster 1 cells in interferon gamma stimulated macrophages are even anti-inflammatory. So we were very excited about this. And then in our pathway analysis demonstrated that in cluster 2 cells, pro-inflammatory factors are highly enriched. And then in cluster 1 cells, anti-inflammatory factors. And also interestingly, some phagocytosis related molecules were representing this molecule, this subset. Then we furthermore isolated these subsets of macrophages, clusters 1 and 2, using cell sorter, then performed proteomics, and results were consistent with RNA, a single cell RNA sequencing. Then cluster 2 cells were more atherogenic. Then cluster 1 cells were atheroprotective. So now I'm going to show you just one piece of evidence, of functional evidence. So this is a efferocytosis assay measured by the device called IncuCyte. Okay, and then green cells represent cluster 1 macrophages. Blue cells, represent cluster 2 macrophages. For example, this guy, green cell, represent cluster 1 macrophages, and then this guy just eats, apoptotic THP1 cells and becomes orange, right? Suggesting that the cluster 1 macrophages are more efferocytotic, which means that the really cluster 1 cells are atheroprotective. But single cell analysis is very interesting. But question is, so what? The very important question is, how can we really translate the biology of cell heterogeneity into the clinic? Then, particularly, we asked ourselves whether we can examine whether, we can shrink cluster 2 and expand cluster 1 macrophages using drugs. Then, another long journey started, and then, we used L1000, drug gene interaction database, to which we input the data from the single cell RNA sequencing to try to identify drugs that can decrease cluster 2 genes, then increase cluster 1 genes. So we identified multiple molecules, including this BI2536, and using this molecule. So we performed additional in vitro validation experiment using single cell RNA sequencing. So simply speaking, in this study, we found that this compound indeed shrunk cluster 2 macrophages and even expanded cluster 1 macrophages. So this is the summary slide of 21st Century Cures Act, and that emphasized the importance of the use of human data, including EHR. That's what we are doing right now. And the idea is the same as macrophage heterogeneity studies. And then we wish to understand the disease heterogeneity using EHR and machine learning. And particularly, we are excited to see the heterogeneity of COVID patients. And then, I'm one of the investigators of NHLBI initiative, and also I'm a member of Recover Omics Task Force. Thanks to the funding from NIH, we are actively working on the heterogeneity of COVID-19 patients, so that we can potentially identify high-risk, subpopulations for long COVID-19, that develop cardiovascular complications. So we believe that biology heterogeneity, is a future direction of cardiovascular research. Technologies, allow us to do something like this, right? We are working on heterogeneity of human vascular cells, and also, as I just mentioned, that we are also working on the heterogeneity of cardiovascular, diseases. Without new technologies such as single cell analysis or machine learning, we wouldn't have been able to do anything like this. Then, from this approach, I hope so we can identify new drug targets for precision medicine in the near future. So these are, contributors to, the data I presented today, and these were former fellows, and then folks that highlighted in yellow color, contributed to the specific, studies I mentioned today. And then these are faculty members, Elena Aikawa is my close collaborator and my wife, and then she's a specialist in experimental pathology and also cardiovascular calcification. Sasha Sin is a director of proteomics of my center, CICS. And these guys are network scientists. And so these guys are very talented microsurgeons. Then without amazing support and help of these wonderful collaborators, we wouldn't have been able to accomplish any of those studies. And then I also thank funding sources, for the support. Thank you very much for your attention.